Hello everyone, it's day 63 and today is Sunday and Sunday's edition. The Hindu has quite good articles over the science page and frequently asked questions page. So today I found this two important articles over this science page in the Hindu newspaper. Today is 5th of May 2024 and the first article is about the can the new organelle help engineer plants to fix nitrogen. So let us see what this article is trying to say. As proposed by Charles Darwin in the 19th century, natural selection, the engine that drives evolution, is how species adapt their to their environments. Unlike the new Darwinist consensus, the American evolutionary biologist Lynn Margulis did not believe that random genetic mutations were the sole cause of inherited variation. She came up with a new theory called symbiogenesis. This endosymbiotic theory states that organelles like mitochondria and chloroplast, the sites of cellular respiration and photosynthesis, were once free living bacteria that were later ingested by the recipient cells. The theory of symbiogenesis was firstly challenged, including Margulis' manuscript, which was rejected by 15 academic journals before finally being published in the Journal of the Theoretical Biology in 1967. So it was not until many years later that mitochondria and chloroplast were accepted as once once being free living bacteria before becoming endosymbionts inside eukaryote cells. So, two papers published recently, one in the journal Science and another in the Cell, have generated new interest in the endosymbiotic theory. The discovery concerns nitrogen fixation. Nitrogen is a key component in proteins and DNA of all living organisms. Although nitrogen gas makes up about 78% of the Earth's atmosphere by volume, plants and animals lack a system that can utilize atmospheric nitrogen. Bacteria and archaea help convert atmospheric nitrogen gas into to ammonia by nitrogen fixation to make nitrogen usable for plants. Unlike many free living nitrogen fixing bacteria, legumes, a class of science in the family Fabaceae, bear the nitrogen fixing bacteria in their root nodules. So ammonia is converted to nitrites and nitrates and then back into the atmospheric nitrogen with the help of bacteria to complete the cycle. In marine environments like on Earth, bacteria and archaea are also involved in ammonification, nitrification, and denitrification. So, beyond mitochondria and chloroplast, the current discovery extend the earlier reports of nitrogen fixing cyanobacterium in marine algae and establishes it as a new organelle. The new organelle that the author will also authors call nitroplast co-evolved with its host cell. So in 1998, Jonathan Zehr at the University of California, Santa Cruz, US, discovered a cyanobacterium candidatus Atelocyanobacterium thalassa or UCYNA in the water of the Pacific Ocean, capable of fixing nitrogen. Later, Kyoko Hagino at Kochi University, Japan, found the marine algae Baru Dosphera bigeloi as the host for UCYNA and could successfully culture the host cell. Both the teams had established UCYN A as a symbiotic cyanobacterium for marine single cell eukaryotic algae. So, bona fide organelles need to satisfy several criteria. First, the organelle must be integrated into the function and overall architecture of the host cell. Second, protein must be imported to the organelle from the host cell to carry out some of its functions. Third, organelles must be in sync with the host cell's growth. Last, organelles must be inherited in the newly dividing cells during host cell division. All these above criteria were satisfied by the nitroplast as presented by the several lines of the evidence by the authors. During a symbionts transformation into the organelle within a eukaryote cell, its genome becomes fragile, encoding fewer proteins and utilizing the host cell's protein to perform some of its essential functions. So, in line with expectations, nearly half of the nitroplast proteins are from the host cell. Although the reports present evidence of establishing nitroplast as organelles, the loss of some nitroplast genetic materials and migration to the host cell nucleus still needs to be established. Unlike mitochondria and chloroplast endosymbiosis, which happened nearly 2 billion years back. Nitrogen's evolution as an organelle is relatively recent, about 100 million years. The discovery has revolutionary implications, especially in agriculture. So, agriculture was transformed in the last century by the discovery of a method for synthesizing ammonia from nitrogen and hydrogen in the laboratory. The current discovery has the potential to play a vital role in getting rid of the harmful side effects of industrial ammonia production. Several novel biotechnological applications may use the result of the current discovery of nitroplast as independent nitrogen fixing organelles. Some they are engineering host cells and their nitroplast with minimum genomes sufficient to grow effectively and fix nitrogen, making plant cells fix nitrogen by engineering them to include nitroplast and organelle transformation in the plant cell to introduce nitroplast and its host genes to fix nitrogen. Although promising and futuristic, all these are highly challenging and far from reality. So, this was a very good article over the nitrogen fixation and ability of plants to fix nitrogen. So let us see one another important article. It is about the H5N1 outbreak, route of transmission unclear. So, what started as a mystery disease in one cattle herd in Texas in March and quickly confirmed as avian influenza H5N1 has now quickly grown to 36 herds across 9 states in the US, with wider testing of the cattle herds ongoing. This number is likely to go up significantly. Two avian influenza H5N1 lineages have been spreading across the world. The influenza A5H1 H5N1 lineage 2.3.4.4.b that emerged in 2022 spread across all continents except Australia, affecting and decimating several avian species. The outbreak has pushed many avian species to the verge of extinction and has spilled over to over 100 mammalian species to date. It is important to note that the recent outbreak in the US is not solely affecting cattle. Farm cats have also been significantly impacted. A total of 7 cats have been documented to be infected in the cattle farms, while there is one documented case of a human with mild symptoms who had close contact with infected animals. The risk appears low. A survey in affected regions in the US found traces of the genetic materials of the virus in nearly 20% of the grocery milk samples using PCR testing. Although further studies have also confirmed the absence of live virus in the samples. However, this survey also suggests that the outbreak might be more widespread than reported cases. Despite this finding, pasteurization, a standard milk treatment, effectively destroys live viruses. Thus, the public health risk is minimal. So, despite the controversy surrounding limited metadata, the US Department of Agriculture (USDA) has provided a large tranche of genomic data for the virus following the outbreak, which has offered valuable insights into the outbreak. So, most significantly, the data suggests that the virus had a single point of introduction in the late 2023 and a potential undetected spread among cattle. A number of mammalian adaptation mutation have been identified in the genomes consistent with the wider spread in the cattle. The data also hints at the possibility of reverse spillover events, transmission from the cattle back to poultry, which differs from previous avian to mammalian transmissions. There are several questions about the outbreak that remain unanswered. Firstly, the route of the mammalian transmission among the cattle herds is unclear, while speculations for this include contaminated feed, milking equipments, or even airborne transmission. Secondly, the question of how the virus evaded detection among cattle population remains open. Finally, the potential of reverse spillover events from cattle to poultry, as suggested by the genomic data, necessitates further research to assess the risk of transmission to humans and develop appropriate mitigation strategies. So, India has seen avian influenza outbreaks in poultry this year across 4 states, that is Andhra Pradesh, Maharashtra, and more recently Kerala and Jharkhand. So these outbreaks have largely been limited to poultry. However, the genome sequences from these outbreaks are not yet available in the public domain, and therefore the lineages of the affecting viruses are not known. Given the fact that the genomic alterations currently occurring in the virus are not adequate to enable human to human transmission, the immediate risk of an outbreak in human remains low. However, close contact with infected birds or animals significantly increases this risk. The transcontinental spread of avian influenza reiterates the need for active surveillance and a much wider collaboration among countries and organizations sharing data and resources for understanding the virus and its transmission dynamics. As the world slowly crawls out of the SARS-CoV-2 pandemic, there has never been a better setting for the global pandemic beauty. So Bani Jolly is a senior scientist at Karkinos Healthcare, and Vinod Sakaria is a senior consultant at the Vishwanath Cancer Care Foundation, who have written this article. So that's all from today's newspaper, and I didn't find any important article over the this page. One important article was about the Tamil Nadu's economy is expected to grow at around 8.08% to 10.69% in 2024-2025 fiscal year. So this is also a good news for the Indian economy overall, because the India will progress only if the states will progress. So let us see if for a few important articles on next day, that is tomorrow. So till then,